Assalamu alaikum girls. How are you and how was the weekend? Mm, Early in the morning in the record करेंगे. So maybe बच्चे भी बीच में उठ जाए. Let's share the screen. Today we are going to study factors affecting the rate of enzyme action. In the previous lectures in the previous week, we talked about what are enzymes and what are their basic characteristics. We talked about anabolism and catabolism. We talked about the mechanism of action, lock and key model, and induced fit model. We started that induced fit model is more acceptable as compared to lock and key model. Then later on, we talked about enzyme specificity, that enzymes are substrate specific. And we also talked about that enzymes are made up of amino acids and how they increase the rate of reaction. And then the active side is made up of amino acids, a short chain of amino acids, which has a three-dimensional shape. So later on we talked about coenzymes and cofactors. Then we talked about uses of enzymes. So kindly keep all those things in your mind while starting this topic. Enzymes are very sensitive to the environment in which they work. I mean, enzymes are not temperature resistant or pH resistant. They are sensitive to any change in their environment. So any factor that can change the chemistry or shape of enzyme molecule can affect its activity. So there are different factors which can change the shape of enzyme and can affect the working of enzyme, and mainly there are three number. Number one is temperature, substrate concentration, and third one is pH. If there is any change in temperature, it is going to affect enzymes. If there is any change in substrate, if the substrate is increasing or decreasing, there is going to be certain effect on enzymes function. And if there is going to be change in pH, again it's going to affect enzymes activity. Either the rate of reaction is going to slow down or is it going to increase? It depends. So there are factors which affect the activity of enzymes, and we discuss them, that they are three number. Let's discuss them one by one in detail. Let's talk about temperature. How temperature affects the functioning of enzymes. Whenever temperature is increased in an enzyme catalyzed reaction, initially the rate of reaction increases. As soon as temperature increases, the speed of reaction increases. But it happens to a certain point, which is called as optimum temperature. Let me tell you about this graph. This graph is plotted between temperature and rate of enzyme catalyzed reaction. On x-axis, we plotted temperature like 10 centigrade, 20 centigrade, 30, 40, 50, and 60 Celsius. Okay, it's centigrade scale. And on y-axis, it's rate of enzyme catalyzed reaction. Initially, it's less, and then as we move towards y-axis, the rate of enzyme catalyzed reaction is increasing. So when we plot a graph between temperature and rate of enzyme catalyzed reaction, as soon as the temperature is increasing, the rate of reaction is increasing. slowly and gradually. You see the red line, it's increasing slowly and gradually. Look at the arrow which I'm pointing towards this graph. The rate of reaction is increasing until there is a certain time where the rate of the reaction becomes maximum. Here in this graph, it is 37 centigrade. I will tell you why. So there is a certain temperature at which enzyme catalyzed reaction will show maximum rate. There is a certain temperature at which every enzyme works at its maximum rate, and that temperature is called as optimum temperature. Later on, when you're going to increase the temperature above that temperature, the enzyme is going to lose its structure. Enzyme is going to change its shape. It's going to fall apart. And as soon as enzyme loses its structure, it cannot speed up the reaction. What is the function of enzyme? To speed up the reaction. But when the structure of enzyme is lost, it cannot speed up the reaction. So the rate of the reaction will fall down, or क्या कहेंगे, will decrease immediately as soon as the structure of enzyme is lost. See, the rate of enzyme catalyzed reaction is decreasing as soon as the enzyme is denatured. I don't know how much you understood, but I'm going to repeat. Basically, when temperature rises to a certain limit, heat adds in the activation energy and also provides kinetic energy for the reaction. I mean, heat is being added and the activation energy is increasing. So the rate of the reaction is increasing. But there comes a time when the heat is increased so much that it breaks the bonds of the enzyme, and enzyme loses its structure. Let's move to the next slide. Initially होता ये है कि when heat adds into the reaction, the vibration of atom of enzyme, बल्कि यूं कहना चाहिए कि enzyme have certain atoms, they starts vibrating as soon as they gain energy. But a time comes that they gain so much energy that the atoms in the enzyme vibrate too much and they lose structure. They break the bond, ionic bond or covalent bond. When they break the bond and lose the enzyme structure, so enzyme will lose its particular shape, and as a result, it will we will call it as denaturation of enzyme. It results in a rapid decrease in rate of enzyme action, and it may be blocked. Um, let me just. And this is a site at which, I think I move to the next slide. So when the structure of enzyme is lost, it is called as denaturation of enzyme. And as soon as enzyme is not there, so who is going to speed up the reaction? So there is no one to speed up the reaction, so the rate of reaction immediately falls down, and even the the whole reaction can be blocked completely because of the absence of enzymes. So this is how what happens about temperature. Let me revise about temperature. Basically, what is temperature? Temperature is a factor which is affecting the rate of enzyme catalyzed reaction. That as soon as temperature increases in every reaction, the rate of enzyme catalyzed reaction increases, but to a certain extent, and the rate of reaction becomes maximum at a certain point, and that temperature is called as optimum temperature. After that, the rate of the reaction can slow down because the heat increases so much that enzyme loses its structure. It is denatured. When enzyme is not there, So how the rate of reaction will increase, rather it will decrease. So ठीक है, after a certain limit, enzyme loses its structure and it denatures, and the speed of the reaction slows down. Now, why the temperature 37 centigrade is mentioned? Because the optimum temperature for different enzyme is different. This 37 centigrade is for the enzymes which work in human body. Because what is our body temperature? In humans, our body temperature is 37 centigrade. Okay? So whatever the enzymes are working in our body, their optimum temperature is 37 centigrade. I mean, कितनी ज़बरदस्त बात है कि इंसान सुबहान अल्लाह कहने को दिल करता है that whatever the enzymes are working in our body, their temperature to work best is 37 centigrade. What I mean to say is that their optimum temperature is 37 centigrade. They show best performance at 37 centigrade. But if it is 36 centigrade, they will show less performance. But it and if it is beyond 37 centigrade, again their performance is going to be lowered, or it can immediately. I mean, it can completely um, slow down if the temperature is too high. But in human body, you know that our temperature does not rise too much beyond 37 centigrade. Otherwise, we are going to die. So in our body, our enzymes are working best at 37 centigrade. So this is all about temperature. Let's talk about substrate concentration. The next factor which affects the performance is substrate. I mean, substrate also affects the rate of reaction. If you're going to add substrate into enzyme catalyzation, the reaction will proceed at a faster rate. Let me just summarize. If enzyme molecules are available in a reaction, increase in substrate concentration increases the rate of reaction. So in an enzyme catalyzed reaction, how many things are there? Enzyme is there, substrate is there, and after a while the substrate is going to convert into product. And okay, so if you're going to increase the concentration of enzyme substrate in a reaction, the rate of reaction is going to increase to a certain limit. But after that, the rate of the reaction will not increase. Let me tell you how. Look at this graph. What I was trying to say, in a reaction, the enzyme catalyzed reaction, the reaction is depending on the amount of enzyme, and it is also depending on the amount of substrate. Here in this case, to check the effect of substrate concentration, we will keep the enzyme concentration constant. Let's talk about the enzyme concentration is 5. There are 5 enzymes in a layman wording, although we cannot count enzymes 1, 2, 3, 4, 5 at molecular level. But let's say, just for our sake, that there are 5 enzymes in the medium, and we are going to increase the substrate concentration from 1, and then 2, then 3, then 4, then 5, then 6, 7, 8, 9, 10, and so on. So if there will be enzyme, then the rate of reaction will increase. But if there is no enzyme, no free enzyme available, the reaction will not increase. Let me tell you one thing. Here we are plotting the graph between substrate concentration on x-axis and rate of reaction on y-axis. Initially, when you increase the substrate concentration, the rate of enzyme catalyzed reaction will increase. The speed of the reaction is increasing to a certain limit, and then there will be a time when the rate of reaction is not increasing. Reaction is happening. I mean, the substrates are converting into product, but they are not increasing the speed of reaction. the reaction will proceed at the same speed. Look at this straight line. Now the reaction is not proceeding at a higher speed. Why? Because there is no free enzyme available. Let me tell you again. When you are increasing the substrate concentration, like 1 substrate, 2 substrate, 3 substrate, initially the enzymes are available. Um, let's say one enzyme and one substrate complex is formed, and then second enzyme and second substrate complex is formed. The rate of reaction is increasing. Then the third enzyme and substrate complex is formed. Third enzyme and third substrate complex is formed. The rate of reaction is increasing. Then fourth enzyme and fourth substrate concentration. The rate of reaction is increasing. Fifth enzyme and fifth substrate and enzyme complex is formed. The rate of reaction is increasing. But now let's say you're adding 6 substrate, but the sixth enzyme is not available. I told you there are only 5 enzymes available in the media. When you are increasing the substrate, like 6 substrate, 7 substrate, 8 substrate, 9 substrate, but there is no free enzyme available. There is no active site of enzyme available. So how the enzyme is going to increase the speed of reaction as there is no active site? So at that time, the reaction is occurring at a constant rate. Let me just share screen on the whiteboard. What I'm trying to say? I'm trying to say that here, let's say there are 1 enzyme, 2 enzyme, 3 enzyme, 4 enzyme, 5 enzyme. Just imagine these are the same shapes. Please excuse my, where's the color? I'm gonna change the color. Okay. Now I'm adding substrate into the media. When first substrate comes, when I add the substrate in the media, enzyme substrate complex forms and the rate of the reaction is increasing. Then the second substrate comes and binds over here. Another second enzyme substrate complex is forming. So the rate of reaction will increase. Now I'm adding another substrate into the media. Again, the reaction will increase because another enzyme substrate complex is formed. So the rate of reaction will increase. Graph is increasing in a diagonal shape. Then when I add fourth enzyme substrate into the media, fourth enzyme substrate complex is forming. The reaction will increase. The rate of reaction will increase. But when I add fifth substrate into the media, fifth enzyme substrate complex is going to form and the rate of reaction will increase. But when I'll add sixth substrate into the media, there is no enzyme available. There is no active site of enzyme available. Let's say 6 substrate, 7 substrate, 8 substrate, 9, 10 substrate. You keep on increasing the substrate, but there is no free enzyme available in the media, so the rate of reaction will not increase. Reaction is happening because there are enzyme substrate complexes in the media, and the reaction is happening at the the reaction is happening at this constant rate in this way. I'm talking about this constant line. Now the reaction is not increasing, rather it is it has become constant because there is no free active site of enzyme available. And this point, the green cross in the graph, is called as point of saturation. There is no free active site of enzyme is available. Because of that, now the rate of reaction is not going to increase, it becomes constant. So this is how substrate concentration affects at the rate of reaction to a certain limit. At the, later on, the rate of the reaction will not increase, it will remain constant, right? I think so. I have clarified the concept quite well. Let's move to the next factor, pH. Okay, pH also affects the activity of enzyme uh, in a very specific way because enzymes are highly selective while in according to the environment, and they're highly selective for their pH. There are certain enzymes which work in acidic pH. There are certain categories of enzyme which work in alkaline pH or basic pH. And there are certain enzymes which are at neutral pH. You should know what is pH scale. All of you are aware of pH scale varies from 1 to 14. From 1 to 6 or 0 to 6, it's acidic media, and 7th is neutral media. And from 8th pH to 14th pH, it's alkalinity or basic media, and the basicity of the solution is increasing, and it's maximum at 14. You know that. So all enzymes work at a very narrow range of pH, and according to that, different enzymes have different optimum pH. There is a certain pH where they work best. The one who work best in acidic media have different optimum pH. The one who work best in alkaline medium, they are going to have a different optimum pH. Okay? I'll give you an example later on. Slight change in this pH causes the retardation in enzyme activity or blocks it completely. Again, the change in pH can change the structure of enzyme, or can block it completely, or can destroy the enzyme completely. Because again, enzymes are very sensitive. Every enzyme has specific optimum pH. Look at this graph. We are going to plot a graph between pH on x-axis and rate of enzyme catalyzed reaction on y-axis. On y-axis, the rate of reaction is increasing. So it's a bell-shaped graph again. And this graph is regarding enzyme which is working in alkaline media, which is showing best performance at 8 pH. Look, this is in the middle of the bell-shaped graph. This is here, this enzyme is showing best performance. As soon as the pH is increasing here, if we come back to the initial point, the rate of enzyme catalyzed reaction is increasing, and it is at its best performance at, it's showing best performance at 8 pH, which means this enzyme is alkaline. This enzyme is showing best performance in alkaline media, in basic media. As soon as the alkalinity is increasing, the rate of enzyme catalyzed reaction is decreasing. So this particular enzyme shows worst performance at pH 8. There are other enzymes which show best performance at pH 2 or at pH 4. Different enzymes are showing best performance at different pH. For example, pepsin. You might have heard this enzyme. Pepsin works in our stomach. Pepsin gets active in acidic media, and the pH in our stomach is It varies, but mostly it's 2. So this pepsin is also showing best performance at pH 2. Pepsin activates in acidic media and it performs well in acidic media and shows its best performance at pH 2. While there is another enzyme trypsin. Why I'm giving an example of pepsin and trypsin? Both these enzymes are digesting protein. Pepsin also digests protein, trypsin also digests protein. But pepsin is active in acidic medium, trypsin is active in alkaline medium, basic medium. Pepsin is present in stomach, while trypsin is present in small intestine. So I must tell you here that we know that in stomach we have acidic media, but you might not have heard that in small intestine the alkalinity is basic. So trypsin shows best performance at pH 8. Trypsin is active in small intestine at pH 8. So trypsin is also digesting protein in small intestine. Right? So I'm just telling you the different enzymes work at different pH, and that pH range is very narrow. As soon as that range changes, they denature, they lost their structure, or they are blocked, they do not work. Change in pH is losing their structure. How? Change in pH can affect the ionization of amino acids at active site. Remember the active site of enzymes? I told you that active site of enzymes is also made up of a sequence of amino acids, and those amino acids are a structural compound, have a certain chemical structure, like they have carbon hydrogen bonds, they have certain bonds. As soon as the pH is changed, some of the ions like hydrogen atom or certain atom ionize. They break their bond and make ions. As soon as a hydrogen ion is formed or a sodium ion is formed or anything ionizes from the structure of the enzyme is lost. The active site of the enzyme loses its structure, so it's not going to function anymore. When you are going to change the shape of enzyme, change the chemistry of enzyme, it's not going to work. So it is by ionization of certain amino acids at active site. And you know what is ionization? The process of becoming an ion. A hydrogen ion is being lost from the structure, or sodium ion or a certain any atom, any molecule is lost. The structure is lost. So enzyme is not going to work. This is all about pH, and this is all about the factors affecting enzyme catalyzed reaction. I talked about temperature, I talked about substrate constitution, I talked about pH. Again, today I woke up for the whole night. If you do not understand, if you haven't understand it well, you can ask me question and I will revise. I feel as if I haven't, but you can ask me question. Let's move to the next topic. The paragraph which I skipped in the beginning from first page, the last paragraph of first page of this chapter. And the question is, how does enzyme lower activation energy? Enzyme lower activation energy in different ways. How do they perform? How do they actually chemically behave to lower the demand of activation energy? How they are lessening the demand from, let's say, 10,000 kilojoules to 2,000 kilojoules? How? There are three main things mentioned in your book, and I will try to teach them because they are complex. These points are complex. I will try to teach you in my own way. Number one, enzymes may alter the shape of substrate and reduce the requirement of energy for this change. Alter the shape of substrate. Let me share screen. Whiteboard. For example, clear. This is the shape of the substrate, and it has to convert into product. And as soon as it is going to convert into product, um, let's say it requires 5,000 kilojoules. But if you lower the demand, the reaction is going to proceed faster. How the enzyme is going to affect? I'm telling you in an easy way that maybe enzyme will change the shape of the substrate, let's say in this way, changing the shape from here. Actually, digitally, I cannot make a perfect diagram, or maybe let me undo it. Let's say enzyme is changing the shape of the substrate in this way. Enzyme has changed the shape of the substrate in a way that it can quickly convert into product. See, कि अगर enzyme ने ऐसे change कर दिया shape को, that less time will be consumed, that a product is formed. यहाँ से मैं इसको क्लियर कर दूँ, यहाँ से products बन जाए, 2 products, one here and one here. So cutting the substrate from here will take more time as compared to cutting the substrate from here. Will take very less time and less energy consumption. So enzyme work in this way by changing the shape of the substrate, and this is how reducing the time and reducing the energy consumption. Acha, what is the other way? Some enzyme do so by disrupting the charge distribution on substrate. Disrupting means breaking the charge distribution on substrates. You can change the word disrupting to breaking or altering, जो आपको easy लगे. Share. Disrupting the charge distribution on substrate. आप तो साइड पे हो जाओ. Yeah, let me just clear. Disrupting the charge distribution on the substrate. Let's say this is a substrate, and now the substrate has to convert into product. Let's say substrate has positive charge and negative charge over here. Positive, positive, but यहाँ पर negative है. Negative, negative, negative. You know that from previous knowledge that opposite charges attract each other. I just want to tell you that there is a, let's say, sequence of amino acids which has positive charge, and here there is a sequence of amino acids which has negative charge. So they are going to attract each other. There's going to be a strong attractive force because opposite charges attract each other. But enzyme may change the charge distribution on amino acids, and somehow it makes it positive positive. Enzyme is making repulsive forces, positive positive on both sides. Somehow enzyme will disrupt the charge distribution on the substrate. Let's say positive positive, like charges repel each other, so there will be repulsive forces rather than attractive forces. What I'm trying to say in a layman's way, that enzyme is changing the charge distribution. Enzyme is removing the attractive forces, rather adding repulsive forces. As a result, the substrate will quickly convert into product and less energy will be consumed. Less time consumption, less energy consumption. The third point was, they may, third point to screen पे होगा. So let's go on the screen. Now here from here. Now the last point is enzyme may also lower activation energy by bringing substrates in the correct orientation to react. Bringing substrates in the correct orientation to react. Orientation means correct angle to react, best angle to react, such an angle that the substrate will quickly convert into product. Let me give you again a very easy example. For example, there are substrates which are, look at these two sticks. They are stick together lengthwise. I mean, there are two sticks, two substrates, which are lengthwise attached to each other. So when the enzyme is going to work on them to convert them into two products, like two arrows, I repeat, this is one substrate and they are attached lengthwise all over, and the enzyme has to convert it into two products. As they are attached lengthwise all over, so enzyme requires more energy to convert them into two products. But if enzymes behaves smartly and changes the angle of these two things and brings it into the 90-degree angle, the enzyme is changing the orientation of the substrates. Now they're attached only at one point. The two things are attached only at this point. Okay? Now very less amount of energy will be consumed to convert them into two products, into two sticks. Right? What I'm trying to say is that enzyme will spend more energy to separate these two sticks which are attached lengthwise completely. It will require more energy. This angle is such a bad angle. But if you change the angle of the substrates, you will spend less energy to convert them into product. So this is what I meant. That enzyme is bringing the substrate in a best orientation. So the substrate will quickly convert into product. As a result, less energy will be consumed. So if I summarize these points, enzymes sometimes change the shape of the substrate. So the substrate quickly converts into product, consumes less energy, activation energy. And sometimes enzymes change the charge distribution. So somehow that it weakens the charge distribution and the substrate quickly converts into product. Again, less energy consumption. Or enzymes changes the orientation of the substrate. It changes the angle of the substrate. So the substrate quickly converts into product by consuming less energy. So this is how enzyme works. This is how enzyme lowers the demand of activation energy and speeds up the reaction million times faster than the original rate of reaction, than the uncatalyzed reaction. So this is how enzyme behaves, and this is almost the end of our chapter. And tomorrow we will do the exercise. I will show some blank screen or something like that, and we will solve exercise. Please ask me questions on WhatsApp. If you don't understand any factor, anything, I'll repeat. So Allah Hafiz from now on. Allah Hafiz.